Gallbladder is act as a bag, it is a storage bag. There is no secretion, there is no bile juice secretion or production here. It just store that. Bile was sent out or collected through hepatic duct and comes into the gallbladder. It stores here for some time. Gallbladder is rich in solids because at the time of secretion in liver, bile juice is rich in water. Solids are low in amount. Here at the time of collection, the solid amount is raised and here stored. Suppose the amount of cholesterol or salts is very high, it leads to sedimentation or formation of gallbladder stone. That is the main problem happens in somebody's gallbladder stone. Gallbladder stone means cholecystokinin. That is a method of treatment that is here. Cystectomy means formation of stones. Cystic means stone. Gallbladder stone is by the presence higher amount of cholesterol and the salts. What are the salts? Sodium and potassium, tarogolates and glycogolates. These bile juice, how much the maximum quantity is? Only 50 ml. This gallbladder seems very dark green in color and its taste is very bitter because of the presence of salts and bile pigment also. This was connected to the main bile duct by a small duct, that is cystic duct. The contraction or release of bile juice from the gallbladder was done by a hormone, that is cholecystokinin. This cholecystokinin was secreted in the duodenum itself, and another one hormone secretin also helping here, but the main control was done by the cholecystokinin. Cholecystokinin secreted in the duodenum of small intestine. This initiate the contraction of this muscles, gallbladder muscles. So it releases the bile juice. There is no enzyme in the bile juice, but the bile juice is alkaline in nature, so it can emulsify the fat. At the same time, it can then neutralize the chyme molecules. So mainly bile act on the fat molecules. Emulsification happens in the duodenum region. Next we will discuss about its functions, how it will be function and where it happen, how in the small intestine region. We will discuss it in the coming slide. The food from the stomach enters into the small intestine through an opening, that is the pyloric sphincter. Pyloric sphincter means contracting muscles. By the pyloric sphincter, it opens into the duodenum itself. Small intestine have three regions: duodenum, jejunum, ileum. Duodenum is the shortest part. Here all the chemical digestion and absorption of food takes place. Okay. The bile juice and pancreatic juice, bile juice secreted in the liver and stored in the gallbladder, now comes through a duct, and at the same time pancreatic juice also comes through one duct and opens into a ampulla of vater. Here, from here only it forms a single duct, that is hepato-pancreatic duct. Hepato for liver, pancreatic to define, it comes from the pancreas. Through hepato-pancreatic duct it opens into the small intestine. Okay, in small intestine most of the digestive process ends in the duodenum itself, because pancreatic juice contains all the proteolytic enzymes and it have the fat digesting enzymes also. So these juices, bile juice and pancreatic juice, acts on the chyme molecules, and the small intestine have some special cells, mainly three main cells and some other cells also seen here. What are the cells? Means goblet cells. We already know goblet cells means a bowl like cells, they secrete mucosa, and very important cell is the paneth cells, which helps in the which act as an antimicrobial agent. It secretes defensins. Paneth cells gives immunity, and Argentaffin cells secrete serotonin, same thing like the enterochromatin cells of stomach. Okay. Now the acid chyme is neutralized. So main function of bile is neutralization and emulsification. Pancreatic lipase starts acting on this fat molecule. So fat digestion initiated here, because already in the stomach itself carbohydrate digestion and protein digestion is started. So now lipid digestion is initiated only in the place of small intestine. And the intestine digestive process will discuss later. in intestine, in small intestine, it have small finger like projection. This finger like projection is called as villi, microvilli or intestinal villi. They are mainly helping for the process of absorption of food materials, absorption of smaller materials. In between on the villi, the base region, it is called as, that groove part is called as crypts of Libercone. It was found or identified by the person Libercone, so it is called as crypts of Libercone. These crypts of Libercone also secrete a product that is totally called as succus entericus. Succus entericus means nothing but intestinal juice or intestinal secretion. These succus entericus also have minor fractions of enzymes that also helps in the digestion of food, and the digested food also absorbed with the help of intestinal villi. This is a structure found in the small intestine. Intestinal villi is mainly mostly populated in the jejunum region. It is found in the duodenum and ileum itself also, but mainly concentrated in the jejunum region. So, major absorption happens in the jejunum part. Okay. And the pancreatic enzyme like trypsin, it is converted, trypsinogen is converted into trypsin in the duodenum region only. Next, chymotrypsinogen into chymotrypsin by the trypsin, that also done here. These enzymes acts on the food by one. First of all, the polypeptides come from the chyme molecule that was breakdown into dipeptide and monopeptide by the help of trypsin and chymotrypsin. Next one, carboxypeptide is breakdown into amino acids. Amino acids are classified into two types. One is the essential amino acids and another one is the non-essential amino acids. Totally 20 amino acids are identified, 10 are essential amino acids. What means essential? We should intake these amino acids from our food. The next 10 are the non-essential amino acids. Non-essential amino acids mean they can be synthesized in our body itself. So, there is no need for no compulsion for taking it through food, but without the help of protein, the amino acids will be synthesized. That needs some proteins. The breakdown amino acids goes to the liver where it is stored, and here only they give or they are used for the production of proteins. These amino acids helps in the production of protein synthesis. Next, carbohydrate, it has some special enzymes, namely maltase, which acts on the maltose molecule. Maltose is the grind sugar, especially found in the grinds only. So, this maltose is converted into or breakdown into glucose, two glucose molecules. Maltose is the disaccharide. Already the food is breakdown into disaccharide. Here it has some enzymes like maltase, sucrase, lactase, which acts on this disaccharides and make down or break down into monosaccharides like glucose, fructose and lactose. First one is the maltose, next one is the sucrose. Sucrose is the cane sugar. This cane sugar is break down into fructose and glucose, glucose molecule. That was done by an enzyme sucrase. What is the specialty of fructose? Fructose is the sweetest sugar which commonly found in the fruits. So, fructose is called as fruit sugar. The next one is the lactose. Lactose means milk sugar. This lactose is break down into galactose and the glucose molecule. Galactose and glucose molecule. So, what are the final products means glucose. Maltose means purely glucose. Next one, sucrose means fructose plus glucose. So, glucose, fructose, galactose are the final monosaccharides produced or formed in our digestive system. Okay. And at the time of DNA and RNA digestion, it gives deoxyribose sugar and ribose sugar. These are also the monosaccharides. These monosaccharides are absorbed in the small intestine region by the help of villi, and they are transported by active and passive transport mechanisms. The fibers alone not at all digested here because there is no enzyme in the name of cellulase in our body. Cellulase is an enzyme helps in the digestion of cellulose. Cellulose is the fiber found in the plant starch. All the herbivorous animals have this cellulase enzyme to digest cellulose. So, because of the absence of cellulase enzyme, the fibers alone sends as waste into the large intestine region, and Fat molecules means small droplets, they are looks like oil droplets, so they can't able to be absorbed effectively by the small intestinal villi. So they enter into the lacteal. Lacteal means inside the villi it have the lymphatic fluid flow. That lymphatic vessel is called as lacteal. This enters into this lacteal in the form of micelles. Then it is reformed into chylomicrons. So it is found as chylomicrons in the blood. That is the pathway of fat. But the remaining monosaccharides and amino acids can be absorbed by the intestinal villi. Okay, we will discuss about the function of large intestine in the coming one. So all the digestion process finished except the fiber matters in the small intestine region. Now in the duodenum, jejunum and ileum, absorption takes place with the help of intestinal villi. How the absorption happens? Absorption means intaking of the micromolecules like glucose, amino acids, fatty acids alone goes and enters into the lacteal by simple diffusion method as in the form of micelle. All the other compounds like carbohydrates and proteins absorbed. How the absorption happen? For carbohydrates, glucose and galactose are absorbed by the method of sodium co-transport mechanisms. How means we already know some co-transport proteins are present in our body. By that co-transport mechanism, especially sodium co-transport mechanism, this glucose and galactose absorbed mostly absorbed food substance is the glucose material. Fructose shows its own way, that is a facilitated diffusion. That is a facilitated diffusion. So the next thing is the protein. Protein is also absorbed by the method of facilitated diffusion and co-transport mechanisms. The intestinal epitheliums, luminar membrane which have the intestinal epitheliums, helps in that absorption and transport of food from small intestine into that microvilli into the blood capillaries. So at the time of absorption, ultimately the food substance enter from the small intestine to the blood capillaries. From that blood capillaries, it goes in the pathway of portal system and ends in the liver itself. In that liver only the glucose molecules are used, and remaining glucose molecules are stored in the form of glycogen in our liver itself. And proteins are amino acids are break down and gives waste as a ammonia, and it is again recreated as special proteins which are needed for our body. And the fat molecules form itself into micelles. These micelles enter the lacteal through simple diffusion method. These lacteals are nothing but oil droplets, small droplets like oil. Oil droplets resembles as oil droplets. They together and form micelles. These micelles laterally enters into the blood capillaries. They are called as chylomicrons. So digestion and absorption comes to an end. Next, absorbed food should be utilized. That utilization process is called assimilation. Assimilation means utilization of food molecules are with the help of oxidation. Without oxygen, we can't able to get energy. That is assimilation process. So this is the end of absorption. That is the end process happens in the small intestine itself. So all the regions of small intestine, whatever it may be, the duodenum or jejunum, ileum, everyone helping for the same function. The next thing is about the large intestine.